lab, I'll do a little mini lecture on what a carbohydrate is, and then, no problem, Paula, you're good. I'll do a little mini lecture on what a carbohydrate is, and then the different tests you're going to be doing. So basically, you're going to be doing a bunch of reactions to determine if you have reducing sugars or not in solution. But we'll try and get through organic acids and bases first here, right? So last time before the exam, we ended with organic compounds of oxygen. We are going to have some more organic compounds of oxygen, and the reason that we kind of separate these is because amines and amides kind of act like bases in solution. Carboxylic acids act like acids in solution. So this is why this is called organic acids and bases. But carboxylic acids, alcohols, amines, amides, esters are all the major functional groups that you're going to see in a lot of biomolecules. Right? Okay, so the two organic functional groups that we have that are organic acids and bases is we have carboxylic acids, which have the form R, which just represents an arbitrary chain, and an OH group here. The organic bases we have are going to be amines, which have an arbitrary group like this, and those are called amines. So those act like bases, which means that in solution, they generate OH minus. Usually amines are going to be protonated, so they can be protonated to form a positive charge. And carboxylic acids, they donate protons, and they're going to be deprotonated in solution a lot of the time. Right? And so these are relevant because they form a lot of the structural components. So carboxylic acids, for example, these form structural components for fatty acids, the synthesis of proteins, carbohydrates, carbs and sugars, and then lipids as well, right? So these have a lot of the carboxylic acid. So if maybe you guys have heard in some of your biology classes that you have an N-terminus and a C-terminus for a protein or an amino acid. If you've never heard that before, so what you're referring to is the C-terminus is the carboxylic acid side of a protein. The N-terminus is the amine side of a protein, right? So this is where you might have heard these terms. And the amine groups are also part of the protein backbone. And they can combine with carboxylic acids to form amides, which are going to be peptide bonds or the bonds that hold proteins together, right? So they can react with carboxylic acids to form amides. And the amides are, we'll talk about, these are responsible for what are the peptide bonds in a protein. Right, so we're going to be focusing on kind of the chemical. So if you've taken a biology class before, what we're going to be focusing on in this class is more how the molecular structure affects biological function. Whereas from a biological standpoint, usually you just look at the function. You're not necessarily worried with the aspects of structure of these molecules. But we know how to draw these molecules, how they interact, the forces that are involved with how they interact with each other. So we're going to look at how that impacts their biological function, right? Because biology, it's a little bit more complicated. We don't have tools at the nanoscale to kind of put things together. Everything has to be kind of put together through interactions with each other and assembly processes like that. Okay, so the first functional group is going to be a carboxylic acid. So this is a hydroxy group bonded to a carbonyl carbon. And so the functional group looks like ROHO. And so these are acidic organic molecules. And they're acidic because they donate protons. And does anyone have any idea right now, just by looking at the structure, where the acidic part of this molecule is going to be? Yep, the right side right here. So this right here, this is the acidic proton. And whether or not you have this proton there depends on what the specific value of a thing we call the pKa is associated with this. All right, and we'll talk more about that probably when we talk about amino acids. And the reason that's an acidic proton is because if you were to remove that proton, you have a minus charge. That minus charge, what ends up happening, can be shared between both of those oxygens. And because the negative charge is shared between those oxygens, it makes it more stable. So if it's more stable, it's in a lower energy state, and so it's happier that way. And so that allows this molecule to be acidic. And certain things, depending on the R group, can affect the relative acidity of this proton, right? So if we change the R group, the acidity changes, right? That means it'll make it more acidic. It'll have a lower pH when you put this molecule in solution. And so I know we've learned a lot of names already. I think, just off the top of my head, we only have carboxylic acids, amines left are the only things I'm going to require you guys to know how to name. And so the naming for carboxylic acid is we have the same rules as before, except the ending, so the E ending is replaced with oic acid, or it can also be ic acid if it has a common name, right? So if we have one carbon atom bonded to the structure, then we would call it, bonded to this, it would be ethanoic acid, but the common name for that is acetic acid, right? So acetone gets replaced by just ic acid instead of oic acid. So you're not gonna have to know to distinguish between the two. We're gonna stick with oic acid for naming of these molecules. All right, so that's how we're gonna name the carboxylic acids. Jeff?
I might be getting a bit ahead, but when does it go to ATE at the end?
ATEs is if you form an ester, which is a derivative of a carboxylic acid, and we will talk about esters because those are very important, or if you go to the carboxylate ion, that's when you have the deprotonated version of this as well. So this is called carboxylic acid, so you have ic acid. If you have it deprotonated right now, you would say that's a carboxylate, right? And so that's the ATE at the ending. But yeah, so that we'll get to that. You guys aren't gonna be responsible for naming esters because they can get a little complicated with all the other rules that we've talked about so far. I'm gonna show you what they are, so when you see it, you know what you're looking at. But I'm not gonna require you guys to name esters. Okay. So if we have a molecule like this, OH, and then maybe something like that. And actually, before I go back, I just want to emphasize, because I didn't say it, this is the highest priority functional group. So even when I've talked about these different priorities, and there's a lot of functional groups you don't talk about, and they all have their own specific priority, accounting for every single functional group that is in existence, the carboxylic acid is the highest functional priority group. And this is because this is usually the largest reactive group, right? This is the most reactive group you can have in chemistry. Okay, so we have this molecule. So take a second, see if you can figure out how to name this molecule. So it's similar to an aldehyde. You're gonna start counting from the carbonyl carbon. So we want to name that, and then I'm gonna write a name down here that you guys can then draw the structure from as well. So if we have methyl noic acid. Let's take it first so you can figure this out. So this is the highest priority functional group, so that goes on 1, 2, 3, 4, 5, 6. Like I said, I'm not going to give you a problem like that on the exam, but I accidentally drew a large group there at the end. All right, so the longest chain is 6, which means the prefix for the parent chain should be what here? Hex, right? So that'll be hex. There's only single bonds, so it'll be ane, right? And then, because we know there's a carboxylic acid, it'll be oic acid at the end, right? So this is the parent chain name right here. And this is identifying what kind of molecule, what kind of functional group it is. It's a carboxylic acid, so it's hexanoic acid, right? And then what substituent groups do we have here? 3, 4, 5, and what are those substituent groups? Methyl groups, right? So they're going to be 3, 4, 5, and then what do we have to put before methyl? Tri, right? Because there's 3 of them, right? So let me just organize that a little better. It'll be 3, and then how do we separate the numbers? Commas, right? 4, 5 dash tri methyl hexanoic acid. So let me write that a little neater just down here. So 3, 4, 5 tri methyl hexanoic acid. Like that's how we would name this carboxylic acid. That's you're going to draw the structure for that, right? So you're going to draw the structure, right? Okay, yeah. See, yeah. That's it. So that's it. These are 2 separate. This is 1, and then this is 2. Yeah. So if you guys have that confusion, I make a lot of mistakes in life, as we all do, right? So point it out, and then I will make it more clear, right? So this is the second. This wasn't the name for the first one. This was the second one. Usually you go both ways. You have the structure, name the structure, and then draw the structure from the name, right? Usually drawing the structure from the name is easier because it tells you exactly where stuff is going to be. You can draw it however makes it most convenient for you. Right, so it says propanoic acid, so I'm going to go draw the parent chain first. So how long is the parent chain? 3 carbons, right? So that looks like something like that, right? We have 1, 2, 3 carbons here. 2, 3. Oic acid means what? There's a carboxylic acid, right? That's always going to go on the first carbon here. So we have a carboxylic acid group. And 2-methyl means there's a methyl group on the second carbon, right? So this is what 2-methyl propanoic acid is. And this is the IUPAC name for this. This is probably called isopropionic acid, if I had to guess the common name for this, right? Because carboxylic acids, they have so many common names for them because they're some of the easiest to separate from other molecules. And because of this, a lot of carboxylic acids were discovered way before we had any idea what electrons were. So there's so many common names for a lot of the carboxylic acids that we deal with. We're not worried with common names. We're just going to be worried with, the systematic way of naming things. Actually, yesterday in my organic chemistry class, I had some students, and they had an alcohol, and it was labeled, 3-methyl butanol. And in the lab manual, it says we're synthesizing isopentyl acetate, right? So isopentyl was the common name. Isopentanol was the common name for the alcohol, and the IUPAC name was different. So they're all asking me, where's the alcohol? I don't see it in the fume hood. Like, I can't get any of the material. So they had the IUPAC name on the bottle, but the lab manual had the common name for the alcohol. All right, any questions on naming carboxylic acids? No? Okay. Am I good to go on to the... It's always, this is the highest priority functional group, right? So if you see a carboxylic acid, it's not a ketone. Even if there's other carbonyls on there, if there's something, we have an aldehyde, we have an OH group, we have this, that. This is a carboxylic acid because carboxylic acid group right here is the highest priority functional group. There's other derivatives of carboxylic acids that you can start to classify. We're not going to go into a lot of them. The only ones we're going to talk about are amides and esters. All right, any other question? All right, so how do we form carboxylic acids? Well, you guys know one way. From an aldehyde, you can form a carboxylic acid. And so how do you form a carboxylic acid from an aldehyde, right? An aldehyde looks like this. All right, how do we get a carboxylic acid from that? We oxidize it, right? And when we're oxidizing, we can have something like potassium permanganate. We break carbon-hydrogen bonds, and we form carbon-oxygen bonds. So this goes to OH and O, right? And so here we formed a carbon-oxygen bond, and we broke a carbon-hydrogen bond. So that was our definitions in regards to organic chemistry, is in terms of oxidation and reduction. And we can synthesize it all the way from an alcohol, right? If we start with a primary alcohol, we have something like ROH. If you oxidize that, this leads to our first bullet point here, which is an aldehyde. And then it continues to get oxidized if we have excess KMNO4, potassium permanganate, to a carboxylic acid. So I know some of these reactions look different, but this is the same reaction for both of these. We're just going through an oxidation process. We're not worried about the specific mechanism for this oxidation. We're just worried about, can we predict, if we start from a primary alcohol, we go to an aldehyde, and then we end with a carboxylic acid. And if you look at every step here and you think about oxidation and reduction, we see that we're forming carbon-oxygen bonds and breaking carbon-hydrogen bonds in each of these cases. This maybe could show up on the final just because the final is going to cover organic chemistry and biochemistry. But this is something that we did on the last exam, right? So you guys had on the last exam, you guys had one whole problem where it was a bunch of oxidation problems of different types of alcohols. Any questions on that? And so just to review really quick, if I'm okay to move on to the next, the kind of general reaction scheme for what this would look like is if I gave you an alcohol, maybe something like this, and I said excess potassium permanganate, predict what that product would look like, right? And whenever we're predicting the product, all we're doing is replacing the functional group, the reactive functional group, with the functional group that it's being converted into. And so take a second and see if you can figure out what the product will be for this. If we have excess, that means that we're going to go all the way from the primary alcohol to the carboxylic acid. If I said, not that much, then we're probably going to stop at the aldehyde along the way. Going to is what functional group again? So it's going to go to aldehyde first, and then a carboxylic acid, right? So the final product is going to be a carboxylic acid because we have excess potassium permanganate, right? So this tells us that this is going to go all the way from a primary alcohol to a carboxylic acid. So what I'm going to do is I'm going to draw all the carbon atoms that we have here first, and then where the alcohol is, I'm just going to replace it with the carboxylic acid. Right, so we didn't have to do anything fancy there. It looks a little weird because it's off to the side like that, but that's perfectly reasonable for drawing the structure, right? These molecules are floating around in 3D space in reality, so it doesn't really matter what the actual orientation of it is, right? I usually draw the carbonyl group facing up, but this is perfectly acceptable, right? And so what I did was I just took the spot where the alcohol was and just replaced it with the carboxylic acid there. And again, remember that line from the carbon represents the bond from the carbon to the oxygen. There is no extra carbon in there. All right. Does that make sense to everyone? How do we feel about that? Thumbs up, thumbs to the side, thumbs down. Thumbs up? Okay. And then some no votes out there. All right. All right, so if you always come to office hours, email me, message me, ask questions if it's not confusing. Or you can ask right now, but. Huh? Huh? Where? The product side? So this one, you're gonna have the OH there, right? Because this is gonna go all the way to carboxylic acid. It's not gonna stop at the aldehyde because we have excess oxidizing agent, right? So the idea is here, the difference between a primary alcohol and an aldehyde is that you need more potassium permanganate to be able to do that, right? So if you have a lot of it in solution, whenever I write a problem like that, I'm indicating to you that you're not gonna see this aldehyde at all. Everything's gonna be completely oxidized to the carboxylic acid. If you started from an aldehyde molecule, then you just go straight to the carboxylic acid. Make sense? Okay. All right. So now we'll talk about the 8 that Jeff was mentioning earlier. If carboxylic acid is deprotonated because it is acidic, it donates its proton to make H3O plus in solution. It forms an anion, and so that anion is going to look something like this. And the reason, again, that carboxylic acids are stable is because this anion really goes between two different structures. It goes between this, we draw arrows, so you might have seen these arrows before. That's just indicating the movement of electrons. And then it goes between that and, these two structures, right? And so because it's switching between these two structures, this is the phenomenon that's called resonance here, and that stabilizes this molecule, this charge on this molecule. Let me redraw this part of the molecule again because this one came out a little messy. All right, so that stabilizes this structure. And so this is called a carboxylate ion. And I'm going to write up here, this is not going to be not tested on this because I'm not going to test you guys on naming carboxylate ions. But the naming for this is, instead of oic acid, you have 8. All right, so you just replace it. So, for example, this is propanoic acid, and this is a propanate ion. And so when you put it in solution, usually carboxylic acids are considered weak acids. They donate the proton and they make the ionic form. But not a whole lot of that happens because they're considered weak acids. So it's still usually, depending on the pH of the solution, it'll be either in its protonated form or its deprotonated form. And once we get to proteins, that'll be more important because that'll tell us about the charge on amino acids, which determines how proteins are going to assemble. Again, you're not going to be tested on naming for these. All right. Am I good to go on? See some people still writing. All right. So esters, these are also, I'm not going to testing you on the naming of these because these get a little complicated. And the functional group for an ester is a carbonyl group next to an ether. And so what it looks like is R double bond O, O, R prime. This is what an ester looks like. And this is important, especially in biology. This is how all of the lipids in your body are linked to their associated alcohol, which will lead us to the kind of the way we can define an ester in terms of its components, right? And so esters are going to end up being synthesized from alcohols and carboxylic acids. And so an ester is defined by a carboxylic acid side and an ester side. And so if we had the functional group, here is the ester, R prime. This is called the carboxylic acid side of the ester. And this is the alcohol side of the ester. And like I said, I'm not going to test you on the naming for these, but the naming is going to be, you name the R prime group for the alcohol, then name the carboxylic acid, and you replace ic acid with O8. And so the important thing I want you guys to be able to recognize is if I look at an ester, can I identify the carboxylic acid side and can I identify the alcohol side? Right? You're not going to have to name it, but the takeaway is to identify carboxylic acid side and alcohol side. And the reason is because in the reactions that involve esters, that's what determines your products. If you do what's called a hydrolysis of an ester bond, you will end up with a carboxylic acid and an alcohol. So if you can look at an ester and identify the alcohol side and identify the carboxylic acid side, you know what the reactants are going to be. Or conversely, if I gave you a carboxylic acid and an alcohol, you could determine the ester that makes. I'm good to go on to the next slide. Is that all right? So here, we have an ester. So we're gonna identify the alcohol side and the carboxylic acid side. I'll write the name, you guys don't have to know how to name it, but identifying the different sides is important, right? So the side with the carbonyl is going to be the carboxylic acid side. So which side here is the, for you guys, yeah, it should be the left side, right? So this should be the carboxylic acid side. And this is going to be the alcohol side. Right, and so if you guys did have to name this, that would be, what's that group called? With the little peace sign right there? Butyl has how many carbons? 4, right? 4. And how many carbons does that have? 3, right? So this is going to be, it'll be 3, so it'll be it'll be prop, sorry. And then is it an isomer or is it just a propyl group? It's an isomer, right? And so does anyone remember what the name of this isomer is? Isopropyl. This is going to be isopropyl. And then the carboxylic acid side has how many carbons? 4. 1, 2, 3, 4. And so the prefix for 4 is but, and this is not an isomer, so this becomes butane, and then OH. Right, and like I said, you guys aren't gonna have to know how to name it. The important part is identifying the carboxylic acid side and the alcohol side. You could, however, have a question that said, here we go through hydrolysis, it splits into a carboxylic acid and an alcohol. What are the names of your products? Right? You would be able to say the names of the products because you know how to name carboxylic acids and alcohols. Okay, any questions on this? Ida? Oh yeah, the last sentence. So you would be able to name the products if this was to go through hydrolysis, right? So
Just said, like, here we go through hydrolysis and it splits into what?
a carboxylic acid and an alcohol. We'll go through that in detail on the next, I believe the next slide. We're going to go through the reverse process first, which is called esterification.
You're saying like you could give a question like that.
Yeah, where it's if I said here's an ester molecule and I put it in acid in the presence of water, what are the products of this reaction? The same way when we did the oxidation, you had to predict the products. You could do the same thing for a hydrolysis reaction. And that's important because hydrolysis happens all the time in the body. Esterification, hydrolysis of amide bonds and esters happen all the time.
And you only say that this is an ester.
Well, you will have to look at the ester and identify the functional group, right? Because this is the only thing that has a carbonyl carbon and a ether right next to it. I know it's a lot of functional groups that are coming at you guys. So if there is one part, or not one part, if you were to only make flashcards for one part of this class, I would say functional groups would be an important one just so you can look at the functional group and identify what the functional group is, right? And so on the next quiz, I'll probably have a question that says, here's a big, gigantic, complicated molecule. You're not going to have to name it, but identify all the functional groups that are on the molecule. Right, so that's a good thing to kind of keep your head there because if you know the functional groups, right, that's the first step in knowing how that's going to interact with other molecules. All right, am I good to go on to the next slide? Hmm?
You couldn't give it like a chart or something.
I'll give you guys one once we've kind of gone through all of them. I've started, I think last time I wrote one before the first exam. Yeah, it was like
It's not like a slide or something.
yeah, it's not a slide that has all of them. Actually, yeah, that's a good idea. I can make a nice compiled list of all the different functional groups. That was the knee, man. All right, I guess I don't need any coffee anymore. I just have rage to keep me awake now. So, all right. So yeah, I'll make a nice compiled list of all the functional groups that we've talked about so far, and you'll be like, wow, that's a lot. And that's like 1/3 of all the functional groups that exist. All right, so how are we gonna make esters? So we can make an ester, like I just said. The way we define the ester and break up its components is how we make them, right? So we have an alcohol and a carboxylic acid. Usually for these types of reactions, you need a little bit of acid presence to kind of catalyze it, or you can have some heat added to the reaction, and that can help catalyze this. The actual reaction of esterification is actually extremely reversible. So the forward and reverse rates happen almost identically. So you can influence which direction the reaction goes just by adding more alcohol or by adding more water to the reaction, for example. And so my lab in my organic lab right now is they're making esters. Right now they're doing an esterification lab. The fun thing about that is that it smells like bananas, right? So instead of the usual acetic acid, it smells like vinegar and stuff like this. We have a banana smell in the lab. It's not very good for you, but at least it smells pleasant. All right, so we need acidic conditions for this to happen. And the R group on the alcohol ends up on the ether side, or the alcohol side of the carboxylic acid of the ester. And so what this looks like, and I'm going to draw this in terms of an arbitrary molecule. So if we start off with a carboxylic acid, HO, and we add it, oops, let me write that, make that red, ROH plus HOR prime. This is going to, in acidic conditions, make an ester.
Every time I do inquiry, I just leave it with my supervisor because he makes a mistake.
Which professor?
Not my supervisor.
Oh, your supervisor. Oh, her name is Esther. Oh, yeah, that is actually a name. But usually it has a T in it, right? So it's a mixture between ester and ether. They actually matter. Interesting. I've never seen it spelled that way. All right, so we start off with carboxylic acid and an alcohol, and we make an ester. There's also going to be another byproduct for this reaction. And so if we look at this reaction and we count all of the hydrogens and oxygens that are there on the reactant side, how many oxygens are present on the reactant side here? 3, right? Because we have 1, 2, 3 oxygens present. And how many are present on the product side? 2, right? Which means we lost how many oxygens? So we lost 1 oxygen. So we lost 1 oxygen. And then what about the hydrogens? How many hydrogens are on the reactant side? That I've drawn, yeah. 2, right? And how many are on the product side? 0, right? So we've lost 2 hydrogens. So what do you guys think the byproduct is going to be for this reaction? It'll be H2O, right? It'll be water. So our byproduct is going to be water equals H2O. So that's going to be our byproduct for this reaction. So with esterification, and what the opposite is going to be, usually if you add water or remove water, it determines if you're going to make an ester or if you're going to split an ester into its alcohol and its carboxylic acid molecules. All right, any questions on that? Am I good to go on to the next slide? All right, so now we're going to do the opposite. So we had esterification was the previous reaction. The opposite is going to be hydrolysis. All right, so this is a hydrolysis. Hydro implies water is going to be involved somehow. We just saw a little bit of how water is involved in this. You still need acidic conditions to kind of catalyze the reaction a little bit. And then because of the name, hydro, that suggests you need what in the reaction? Water, right? In the previous, we saw that it was a product of the reaction. In the hydrolysis of esters, it's going to be a reactant in the reaction. And so we're going to start from an ester. R prime. We're going to add water in the presence of an acid, most likely, H plus. And we're probably going to create what two molecules here? We're going to be using this hydrogen and oxygen right here to split this molecule into two. And so what two molecules do you think are going to come from this ester? Carboxylic acid and an alcohol, right? So this is going to be ROH plus HOR prime. Right, and now we started with 2 oxygens, we ended with 3. We started with 0 hydrogens and ended with 2, which means that water was involved somehow to protonate this hydroxy group here and then create the hydroxy group for the alcohol as well. Right, and so these ones, we split up, we start from an ester and we make an alcohol and a carboxylic acid. And these two, you can name. All right, so you can name both of those molecules. So if I give you an ester, it's not important to be able to name it, but you should be able to identify the carboxylic acid side and the ester side, or the alcohol side, and then that tells you how the hydrolysis of this molecule will work. Okay, so let's look at how this can work for a couple different cases. I'll give you 2 reactions, and I want you guys to think about how to predict the products for this reaction. Am I good to go on to the next slide? Good? Okay. So the hydrolysis of esters, let's say we start with that. It's in the presence of acid and water. And then we also have another reaction All right, so predict the products for both of these. You know, I'm not asking you to name anything here, so don't worry about naming anything. First one. So what's the alcohol side and what's the carboxylic acid side? Is this an ester we have here? Yep, is this an ester because we have the ester functional group. Right, and then what's the alcohol side? The right side for you guys, right? So this is the, this is the alcohol side. And this is going to be the carboxylic acid side, right? The COOH is usually how people abbreviate carboxylic acid side. Right, and so we're doing hydrolysis of this ester because it's in the presence of water and H+, which means our products are gonna be what 2 molecules? The carboxylic acid, which is just going to be the COOH, the carboxylic acid side of our ester, and the other molecule is going to be an alcohol, which is just the alcohol side of our ester. And all we're doing is just replacing where the single bonds to the O are in the middle of the functional group with OH for the carboxylic acid and OH for the alcohol. So this goes to OH plus HO, and then we have 1, 2, 3, 4 carbons bonded to it. Okay, does everyone see how the molecule hydrolyzed like that? An easy way to do this is we just take the part with the carbonyl that I have circled in red, and we just add a OH to that. We take the part that I have circled in blue, and we just add an OH to the left side of that. All right, so we added an OH here, and then we added an OH there, right? That's how this molecule splits, right? We don't worry about this oxygen in the middle. We just add an OH, and then add an OH, and then split those two groups apart from each other. All right, what about the second reaction? Is this going to be hydrolysis or esterification? This should be esterification because we're starting from two smaller molecules and we're making one larger molecule. We have, which one is the carboxylic acid? The left side, this is our COOH. Right, COOH again is to abbreviate carboxylic acid, and this is our alcohol. So those are going to come together to form an ester, right? And so we're putting the carbonyl in red, O, another bond, and then the alkyl group here. So this is going to be an ethyl group there. Right, so all I did was I just combined the part I'm circling here with the part I'm circling here, and I put an oxygen between those two groups, right? So these get combined with an oxygen between both of those, right? That's how that esterification is happening. And we end up with an ester. How do you guys feel about that? I see some concerned faces right now. All right, so what's I guess confusing, or where's what doesn't quite make sense here? More practice? Or I can give you guys some practice. It's just the identification of the alcohol or the carboxylic acid side and the alcohol side, and you're just snapping them together with the oxygen in between, right? So identifying what I have in red and blue here, that's the important part. Once you can do that easily, then it just comes down to understanding that this carbon right here, this carbon right here and this carbon right here are going to have an alcohol between them when they come together, right? In the opposite process, this carbon right here and this carbon right here, this bond is going to break, and it's going to get an OH group, and then this is also going to get an OH group right there. So I know the lines are going in a different direction than maybe you're used to seeing, but the ester bond is broken, and you form the two parts of the molecule. Okay. Yeah, I'll give you guys another example. I'll give you two more of each of them, or one more of each of them. Plus H plus, and then Oops, I did the wrong thing. All right, so what kind of reaction is the first one? Is it going to be hydrolysis or esterification? Esterification, right? We're going to be bringing the carboxylic acid. So this is going to be our carboxylic acid. And this is our alcohol, right? Which means what's going to happen is we're going to lose the OH groups here, and they're going to be bonded to each other. This carbon right here is, this carbon is going to be bonded through an oxygen to this carbon right here. So if I start drawing that on the other side of this molecule, O, that's the bond to the oxygen that I was just talking about, and that's going to be bonded to, again, this carbon on the alcohol. And so we get that. Right? If I highlight the carbons that are involved, it would be those carbons right there. Those are all the unique carbons. All right, and what about the second one? Is that going to be hydrolysis or esterification? That's going to be a hydrolysis reaction, right? And so we're going to split it up into the carboxylic acid side and the ester side. So which side is the carboxylic acid side here? On your guys' side, is it going to be on the right of the ester or on the left of the ester? The right, right? Because we have the carbonyl group in the COOH. And then we have the alcohol group that's bonded to the other side of the ester bond, right? And so this is going to split up into both those molecules. So I'm going to draw this molecule and this molecule as is, and then add the hydroxy groups where I need to. And that's why we have water, right? Because we're adding water in the reaction because we need 2 hydroxy groups and we only have 1 oxygen there right now. So this is going to go to OH plus OH right there. Without the hydroxy groups, these are the 2 alcohols, or the 2 sides. This was the carboxylic acid, and this was the alcohol side of the ester. Then we just add the hydroxy groups to where they were connected by the ester bond. All right.
Would it matter if I put the OH on either side?
On either side, you could put the OH on any point of these molecules. If you rotate this one, it would be the same molecule, 360 degrees, 880 degrees, it would be the same molecule. Yeah?
So for the esterification, we're just blending them together with oxygen,
Yep.
and then for the hydrolysis, we're separating with OH.
Yep, we're separating them with hydroxy, yeah. And that's why I said the important part is just to identify the carboxylic acid side and the alcohol side of the molecule, because that's what's important for hydrolysis and esterification, which are important for organic chemistry, but also for biochemistry, because these are important for a lot of the biomolecules we're going to be looking at. All right. Any questions? You guys feel better after the second example? All right. All right, so we'll go over amines real quick, and then we'll take a break, and we'll come back and we'll start doing the carboxyl or the carbohydrate lab. So the amine as a functional group, and these are bases, right? Which means that they create OH minus in solution, and they add H plus to become positively charged. So ammonia starts as NH3, something like this, and when it gets protonated, it creates OH minus in solution. like that, and becomes positively charged. Here I just have hydrogens, but these in a amine as a functional group can have R groups at all of these positions. So that's why we call them bases, because they make OH minus in solution. They take protons from water. And the functional group is going to have a nitrogen at the center, and then potentially up to 3 R groups, right? And so each of these R groups can be a hydrogen or an alkyl group. And the classification of amines, so they get classified like alcohols get classified primary, secondary, tertiary. So we have 3 different scenarios: primary, secondary, or tertiary. This is determined by the number of alkyl R groups. And what that means in a not so jargon way is just the number of carbon-based groups that are bonded to the amine. So alcohols are classified by the carbon that the hydroxy group was bonded to, right? Amines are going to be classified directly by the number of carbon groups that are bonded to that amine, right? So it's slightly different because it's directly related to what's bonded to the nitrogen, whereas alcohols were based off of the carbon that the hydroxy group was bonded to, right? So a primary amine, all of the amines are going to have nitrogen in them like this. A primary amine will have 1 R group and 2 hydrogens. A secondary amine will have 2 R groups and 1 hydrogen. And a tertiary amine will have 3 R groups and no hydrogens. In those R groups, they can be different or they can be the same, right? So you can have 2 methyl groups, or you can have a methyl and an ethyl group, right? These and how you name amines, we're going to talk about in a second, are is related to those specific alkyl groups that are there. So we're only going to be naming very simple amines in this class. Good. Okay, am I good to go to the next slide? All right. Okay. Okay, and so the naming for amines is just going to be naming each alkyl group in alphabetical order and adding amine at the end. So each alkyl group means you naming them as a substituent group. So an example, we have this amine. Draw this a little better. that, we have 1, 2, 3 different groups. So if you were to classify this amine, how would you guys classify it? Tertiary, right? And then we're just going to list them in alphabetical order. So we have a propyl group, an ethyl group, and a methyl group, right? So we're going to write ethyl, methyl, propyl, and then we just add amine at the end. Right, so amines, in my opinion, have naming convention that kind of leads to whatever the name equivalent of a run-on sentence would be, right? Because if you have a tert-butyl, an isopropyl, and maybe a sec-butyl, then you have tert-butyl, sec-butyl, isopropylamine is going to be the name. You just kind of list all of them together. So then the naming convention for amines is relatively simple. It's just knowing the different groups that are bonded to them. Okay, all right, so let's stop there for now. Let's take like a 20-minute break or so, and then we'll come back and we'll start the lab on carbohydrates. And then we'll finish up the rest of organic acid bases on Monday and then get right into carbohydrates after that. I'll give you a lecture on carbohydrates so you know what you're doing for the lab today, but then we'll cover it in lecture again on Monday, or Tuesday, sorry.